Alright, so this is my presentation on the copperheads. This is going to be over the two species we have here in Texas, and uh, depending on which literature or who you ask, I still lean on the side of recent findings that these are the only two species, that being the eastern copperhead, which is Achistrodon contortix, and the broad-banded copperhead, which is Achistrodon lactensis. So anyways, let's get quickly into it. So we're going to start just with the basics of, what is a pit viper. So here we have one of the native species in Texas, at least in East Texas, and this is also one of our protected species. This is timber rattlesnake, Crotalus horridus. They're one of the largest rattlesnakes in Texas and even in the states, I believe, outside of the western diamondback. And obviously the eastern diamondback is also a very comparable snake in length. So right here we have Crotalus atrox, which is the western diamondback. As far as the timber rattlesnake go, it really ranges from about East Texas all over the most of the eastern United States and even into some northern states. As far as the western diamondback goes, you really won't find it here in East Texas. You will find it in central Arkansas. It has a little blip where there are strong populations there. Uh, but as far as Texas go, you'll start finding them pretty much as far east as Cook County, closer to Dallas. And as you head into central Texas, sort of northwestern Texas and North Texas, they become more common. And of course, down in South and West Texas, hence the name, they're very common. Anyways, what pit vipers are named after is the pit. And this can be differentiated. If I come grab the laser pointer down here, this is their nostrils. Of course, they still use this in conjunction, obviously, with their tongue to collect information around the area and send it to the Jacobson organ to sort of be processed in a way. Uh, so, the pits are a little ion membrane channel, and they can sense differences in infrared heat. And this goes, as far as to sense a 600th of a degree Fahrenheit. And that happens to be very useful for them. I'd imagine that given that any sort of biological animal, regardless being, endothermic, like, warm-blooded, or ectothermic, like cold-blooded, during biological processes and whatnot, they will slightly shift temperature. I mean, I believe the human body sits around 97.6 or somewhere in that area. Of course, it's never going to sit perfectly at 97.6. It's going to change to maybe 96.5, 96.8. To them, that is a massive change, and that allows them to distinguish something living between something not. Even in cold-blooded animal, they're not going to be exactly the temperature of their surroundings unless they are dead, so they will be slightly above that temperature. This also allows them to hunt at night. Uh, some vipers prefer to hunt at night, and especially during warmer months, when daytime temperatures are high and it could pose danger to the health, they will become nocturnal when it comes to foraging. So anyway, so this just goes over more or less what I've said, in case this is something you want to, visually see to take notes over. So this is another one of our native rattlesnakes. In the Crotalus genus, we have two genuses of rattlesnakes. We have Crotalus, which is when you generally think of rattlesnake is what it is, and we have Cistrurus. I'm not sure. I think I have a picture of one towards the end. That's going to be your Massasugas, and that's going to be your pygmy rattlesnake, which is a rattlesnake we also have here. Uh, so Crotalus viridis, this is a snake that you're going to find more in central North Texas, and obviously more prairie land. I do believe it will extend into Oklahoma, but it has a somewhat small range compared to some snakes. Something people talk about vipers, it's not exclusive, of course. When you have other snakes, especially Nerodia snakes and Thamnophis, that will, Nerodia being water snakes, and Thamnophis being garter snakes and ribbon snakes, both of which are colubrids. They're both non-venomous snakes, and they will flatten their heads out in the same manner when threatened. Uh, Especially with water snakes, that can make it somewhat difficult from a distance to get close, and water snakes have their own defining features over, say, a cottonmouth. Anyway, so this head has its reason for being shaped like it. If we go ahead and get this over here to the side. So One of the reasons is, and I'll go through and label this here in a second, you have the venom glands, which are large glands that both create and allow the venom to be delivered. And this is done by the compressor glandulae muscles, which are right behind it. If you've ever seen videos of somebody milking a snake, what you'll notice is the snake, they usually have to force a snake on to bite that piece of plastic at top of the collection container. You may notice either they do not inject any venom or they inject some. I've seen a few where they do dump it all, but that's very rare. But generally, what has to happen is the person milking it, you'll see them rub the venom glands to allow more venom to come out. So basically, taking action in place of the compressor muscle. And on the back of the head, the reason it sort of bows out like that is vipers have very large adductor mandibulae muscles, both muscles in that group being the externus profundus and the superficialis. This is just to help them again with envenomation and later consuming the prey. Alright, so if we bring them down here and look at it, again, I'll quickly label these. You have the venom glands, and then again, right next to it, you have those compressor glandulae, and then you just have that adductor group in the back. So this is like one of the only non-native snakes that I've really put on here. This is an African bush viper that I photographed, and I want you to notice something. If you notice, his fangs are not like out; they're sort of folded against his mouth. Uh, of course, this lives in Africa, I believe Central to East Africa, Atheris squamigera. Anyway, so vipers, like Elapidae and some Colibridea, they all have hollow fangs for animation. Of course, there's more snakes. You have boas, pythons, blind snakes, which everybody forgets about. There's all sorts of small families, but of course, vipers, elapids, and colubrids can be your major families. Uh, as far as vipers go, their fangs are highly unique. They have selenoglyphus fangs. Glyphus to have, I guess, fangs in this case. Uh, and this was estimated to have evolved around 35 to 25 million years ago, looking at vipers in the fossil record. Uh, these were the most recent fangs as far as evolution goes. We can track rear-fanged colubrids, that being colubrids like the hognose snake, Heterodon genus, uh, to have evolved their fangs about 65 million years ago, uh, 50 to 65 million years ago, close to around the extinction of dinosaurs. Of course, fangs are hollow on the inside, and the canal connects to the venom gland. And again, the compressor will compress the venom gland, allowing the venom go through the canal and drip through the fangs. And back here again, we have a pisthologlyphus, which are going to be just enlarged teeth in the back of the mouth. Uh, as far as the presence on the venom gland, I don't know at this moment. I haven't looked too much into hognoses, whether they have a dedicated venom gland. But as far as a lot of quote unquote venomous colubrids go, generally the venom components are in their saliva. So this is possibly just something that allows the saliva to get into the prey or a predator faster. Now, as far as elapids go, so think coral snakes and taipans from Australia and the Australasia area and cobras, they're going to have protoglyphus fangs. And what those are is they are front, sort of like the vipers, except they're fixed. They, sort of like our teeth, we can't fold our teeth, and neither can, sorry about clicking on this next one, uh, neither can the elapids. But they do have dedicated venom glands, just like the vipers do, or at least in this case, Viperidae, is the name I'd intended to put. And again, as I mentioned, if you look down here at the African bush viper, you can see his fangs are folded flat against his mouth, uh, and they can swing forward. I've actually put the wrong muscle here. Seems to be a lot of debate, and it's very difficult to find the correct muscles. There's a lot of arguments, and perhaps it changes by snake and by family. From what I have understood reading some recent research, it seems to be the medial pterygoid is what is responsible for swinging the maxilla up. So they're able to actually move their maxilla bone upwards, which is how they erect their fangs by choice, by the way. As you can see with him, he has chosen not to erect his fangs and given something like the cottonmouth, Echistrodon piscivorus, or Echistrodon canadi, the Florida cottonmouth. They're, commonly known display, and what they're named after is in defense, they will wrap up and sort of show their mouth. They will sometimes choose not to erect their fangs, or sometimes when trying to seem more threatening, they'll sort of like make a light strike and erect their fangs, or whatever it is that they choose to do. So here we're going to focus on Echistrodon. This is a broad-banded copperhead right here. We'll talk more about their range in a second here. So all of your pit vipers are going to be found in Asia and the Americas. There are other snakes that are just called vipers that do not have the heat-sensing pits. Sort of like in Europe, you have some species of adder who do not possess the pits that pit vipers have, and these are more than likely the oldest vipers as far as evolution goes. Granted, that term is relative, as recently evolved or whatever goes. Take something like humans and chimpanzees. Seeing as the split from our last common ancestor is estimated to be around 6 to 7 million years ago, you would think that humans are quote unquote more evolved than chimpanzees, which is not true. As far as we're aware, they are a somewhat newly evolved species as well, because I believe the oldest found fossil linked to pantroglodytes, the chimpanzee, is, I believe, 150,000 to 300,000 years old, which is around the same time period as the emergence of Homo sapiens and Homo sapiens adultu. Anyways, back towards this. So if we just get rid of that, we're going to talk about Echistrodon. That is going to be our genus that includes copperheads, which we'll go over soon. So if we bring this over here and blow this up, the first thing, obviously, is the eastern copperhead, the one that everyone is most familiar with. As I mentioned, this is actually the most widely distributed member of the Echistrodon genus. They range, close towards the desert, about sort of central West Texas area, possibly due to some intergrade, all the way through the Midwest. They range into the North Panhandle of Florida, but do not go into South Florida, and they go all the way up the East Coast, sort of. And I believe there's even a few in New York. Now we have the very closely related sort of sibling of the eastern copperhead, which, as I mentioned, is the broad-banded copperhead, Echistrodon latistus, however you want to try to say that. This snake is a little more picky on its range, smaller range. Think South Texas, Central Texas. We're going to range into Oklahoma and all the way up into about north central Kansas. The snake likes deserty areas, it likes prairies, just things that are generally more dry is what I've seen from range-wise, is what it seems to like. Previously mentioned, we have the northern cottonmouth. It has a large range similar to the eastern copperhead, but not quite as far north. It prefers, your more wet habitat, but it can survive, obviously, outside of it. And, of course, as you go into Florida, what we've discovered somewhat recently is that is a separate species of cottonmouth, the Florida cottonmouth, which is Echistrodon canadi. Both of these, the two species of copperhead and two species of cottonmouth, they're very, very closely related, and their divergence has probably only just begun, more than likely because of allopatric speciation. You have two populations of individuals who no longer breed. And so as they change, not necessarily to better fit their environment, but the ones who live to pass their genes, they will become slightly different, and possibly in several 100,000 years we'll have very definite new species, but that is not something we will live to see unless something crazy happens with medicine, in which case I'll see y'all then. So some lesser known members of the Echistrodon genus, we start with actually a very well known member, at least locally, and a very widely distributed, is the Mexican cantil, which is Echistrodon bilineatus. This species, I believe, ranges a little north of Chihuahua and all the way down close to Costa Rica, actually. Very well distributed species. We also have the Taylor's Cantil. It's a little more specialized, I believe, into the east coast of Central America and the Mexico area. We have the Gloyd's moccasin, which is a very small sort of cantil, Echistrodon howard-gloydi, that is pretty much only found in Costa Rica. And unfortunately, this is one I couldn't even find an image of. So this is a creative interpretation by artificial intelligence using the like one or two photos that realistically exist of this snake. And we have the Yucatanian cantil, which is Echistrodon raceolus, which is found, of course, on the Yucatan Peninsula in Central America. Sorry. So here in Bowie County, we have two species, certainly two species. We have Echistrodon contortix, and we have Echistrodon piscivorus. Now, as I'll go into, we could possibly have Echistrodon lactinctus, the broad-banded copperhead, or at least intergrades of it, but it has never really been confirmed, so that's not something we know at this moment. Alright. So just a quick little glance at these two. So you have the eastern and the broadband. As I had mentioned before, the eastern copperhead is very widely distributed between the southern and eastern United States, while the broadband copperhead is more into central, west, and sort of south Texas, into Oklahoma, and southern to centralish Kansas. The eastern copperhead tends to be more of a generalist in habitat and diet, whereas, as mentioned before, the broadband prefers its more sparsely foliated areas. We will go over patterns in a little bit, so don't worry too much about that. On average, these snakes only get about 2 or 3 feet in length, compared to the cottonmouth, which almost can get twice that size, and rattlesnakes that, again, usually get twice that size. They're a medium-sized viper. As mentioned before, they can hybridize in those intersecting range, which is called an intergrade, and it makes, trying to identify between the two in these intergrade ranges is just so fun, obviously without genetic testing, as you can have patterns that mix, and realistically, outside of a few scale differences, pattern and location is pretty much the only defining feature. And they can hybridize with other members of Echistrodon, which I'll get over. So here we have an eastern copperhead. In the past, there was considered to be several species, or at least subspecies, of Echistrodon, and a lot of snakes and vipers as well. This has changed as we've done science has advanced, and we've done more genetic testing. Be like, oh, these are significantly different animals, and they're not the same organism or even a subspecies. In the past, this consisted of the southern copperhead. I believe they even had an eastern copperhead, which is now what has been lumped into. Being that the eastern copperhead was Echistrodon contortix, the southern copperhead was Echistrodon contortix, which is done with slightly different species going back to humans. Modern humans are generally considered to be Homo sapiens sapiens, given that we have changed slightly from Homo sapiens that were found, say, 150, 200,000 years ago, anatomically and somewhat genetically, especially as we interbred with Homo neanderthalis, the Neanderthals, and possibly other species. Again, too far off the tracks there. There was also other species that were considered at the time, such as the broadband still. You had the Osage copperhead, which I do believe was Echistrodon contortix, one of the subspecies. And we have the Transpecos copperhead in sort of that West Texas area, which was considered a subspecies of the broadband. Anyways, what makes contortix unique is sort of that just sort of Hershey kiss look that they have to their bands. These are going to be small bands that generally narrow at the top, and on the belly they're going to have sort of these spots that continue. So here we have a broadband. As you notice, instead of having that Hershey kiss, of course, we still have some of the bands in arrowing, which again in intergrade ranges gets a lot more difficult to tell. But anyways, as you notice, these bands sort of stretch straight and all the way over, and they sort of have this rectangular and trapezoid. And as I mentioned, broadband was also considered a subspecies of Contortix, and again, the subspecies list just went on until eventually someone tested genetics and went, yeah, these are the same snake. They just look a little different because they're in a different area. Uh, as I'd mentioned, they have these broad bands over them, and a lot of times the bands will also extend onto their underside. Here we have a newly hatched one. As you notice, he has the Hershey Kisses, so we're going to assign him to the eastern Copperhead. Obviously, the very big detail with this snake is that the tail is bright yellow. There's a lot of snakes that have this. I believe Massassaugas in the Cistrus genus, they have colored tails. And obviously all other members of Echistrodon, they have colored tails, which is sort of a yellow to chartreuse color. And the reason they do this is that they're able to use it to lure small prey like insects, amphibians, or other reptiles that may be tempted to see, oh, there's a bright little thing over there moving, I gotta go get it. Uh, Generally, after a year or two of age, once they've had a few shed cycles, that color will fade away and they'll match the body. Although I have seen pictures of, 2 or 3 foot long adults that still either sort of have it or fully have that color to them. This funny looking thing is the Cottonhead. What this is a hybrid between the female Copperhead and the male Cottonmouth. And of course, you can reverse it, and they sort of nickname it the Coppermouth, but generally it is still called the Cottonhead. Uh, since, as mentioned, all North American Echistrodon are related, their hybrids are also fertile, since they're not that well diverged yet as a group of species. Possibly, it is possible that some members of North American Echistrodon could breed with, say, the Cantiles, but no one has ever tested that. But if you're looking for a grad school thesis, that might be a really nice one. Anyways, this is a photo I took of one from Miss Brooke Howell, and something you'll notice here, if I grab my pen, is did not get a chance to go over some defining features of the Cottonmouth over the Copperhead. First thing I'm going to look at here is the head. So first of all, you notice that the eye is sort of glossed over, sorry. Well, all that means is that the snake is very soon to shed, and one of the things they do shed is that sort of scale over the eye as well, since they do not have eyelids, unlike lizards or a lot of other reptiles. So a big thing about copperheads is they do not have this band behind the eye. I've heard people call it the Zorro mask. This is something that is unique to cottonmouths as opposed to copperheads. What you can see is you do notice that sort of Hershey pattern, but instead of looking that sort of smooth and cream color, you'll notice it looks real pixelated, which is also another defining feature of cottonmouths. Additionally, here we have the bull's eyes, but again, you also have several other features found in the copperhead. Again, as mentioned, you have those sort of Hershey kiss colored patterns, shape patterns. That Zorro mask, instead of staying all the way dark, you still have it lighten up around the top of the mouth, as the copperheads have. And as pretty much as far as the features go, there's some scale differences that are very small and minute, but that's pretty much just going to be it. And of course, obviously, as mentioned, all the hybrids are fertile, so the hybrids can continue breeding with each other. Here they tend to still retain the cottonhead traits, but of course, if bred with copperheads or cottonmouths, they slowly water back down to one of those species. The hybrids have only been documented between the eastern copperhead as well as the cottonmouths, the two species. As far as I'm aware, no one has documented a broadband with the two, and obviously no one has documented Cantiles with other Cantiles or Cantiles with North American Echistrodon. As far as something I've heard a lot of people say about the cottonhead is, why would you breed that thing? We don't know anything about their venom. It could be dangerous, but more than likely, as far as we're aware, crowfab or any other anti-venom that is used for pit vipers in the U.S. will probably work on this as well. So, just some habitat facts about the two. Just a small attention to detail here. If you're ever writing a scientific name, this is capitalized. This is not capitalized, so make sure you catch small things like that. Echistrodon contortix is present in Bowie County and the county surrounding it. Of course, Arkansas and Louisiana. It goes a little far west, but again, as soon as we start getting into Cook County, that county close to Titus and Dallas, you'll stop seeing them and it'll transition into broad-banded copperheads. And of course, as I mentioned here, the theoretical intergrade range goes as far east as Cass County, which makes sense, as I'll get into in a little bit, because of some features found in Cass County. Now, majority of Bowie County is the Piney Woods ecoregion found in East Texas. This is an ecoregion that is going to be obviously predominantly loblolly pines. You're going to have fans, both invasive and native. A lot of rainfall, a lot of creeks and wet areas, very biodiverse area as far as everything goes, even down as small as something like ants. Of course, you will have some pines, think water oak, pin oak, some post oaks, red oaks, of course. Now, Lectinctus, as we mentioned, is a little more picky and they like their open habitat. The west side of Bowie County, we're going to start transitioning to post oak savanna and prairies. We're also going to have, again, like blackjack oaks and post oaks that start it. And again, as anyone who's ever driven on the interstate towards Dallas, you'll notice we'll go from this sort of closed off dense woods to just more open fields and sort of dense collections of, very small pastures of trees. So, as far as they go, just 1 second here. During the spring and most of the year, copperheads will be diurnal. They'll be active during the day and sort of be inactive during night, more or less. And obviously that is because, especially here in East Texas, when, it become June, July, especially August, it just gets hot. And, as animals that are ectothermic and cannot regulate their temperature as well as something like mammals, that tends to be deadly towards them a lot more so than us. So during the summer, copperheads and most other snakes, they become nocturnal. There's another term I can't think off the top of my head. It's what deer are. It's an animal that generally moves during low light hours, such as dawn and dusk. Snakes will also tend to follow this, but will mostly be nocturnal, just due to temperatures, especially in sort of the AM hours where your solar minimum occurs and you have the lowest temperatures. But yeah, they'll become active at night to forage, to do a lot of things just so they can avoid the heat. And of course, as winter comes on, winter of any sorts, doesn't matter if it freezes or not, they're going to brumate, which is a reptile's version of hibernating. Instead of going to this deep sleep, they just become less active, just spend a lot of the day just sitting. Of course, they will come out to drink, to eat once in a while, and to bask, obviously, to build up their heat on warmer winter days. But for the most part, they're going to be inactive, and they're just going to find hibernaculum, crevices, stumps, burrows, or just anything they can sort of get out of the cold. And they do habitually return to the same burrow every year if they can, and a lot of times these burrows, if they have one copperhead, they have a lot. As I'm going to also get into with the next point, they also overwinter with a lot of other species, think rattlesnakes, cottonmouth water snakes, even king snakes. This is not really, as far as nesting with other copperheads, they're fine with it. As much as an animal that cannot process emotions like that is fine with it, but otherwise, when it comes to nesting with rattlesnakes, cottonmouth water snakes, it's more of like a tolerable thing. Like hibernaculum is obviously limited in some areas, so if an area is good for one snake, it's generally good for a lot of them. I'd imagine once they become more active, they probably don't like each other and start eating each other, but good for them. So as I mentioned again, the copperheads will, or just any snake, will bask on a warmer winter day for short periods of time just to bring their body heat back up. Here we have two broadbands in just a little stump, and they're just sort of just hanging out there. If they're don't have something to do, like to eat, drink, or bask, they're just going to sit there all day. So we have two broadbands right here. Uh, Now, for the most part, copperheads aren't terribly social, which I think I'll get to at the end of the slide. Lots of pit vipers tend to be less social than something like Thamnophis garter snakes, which will have dens of hundreds of snakes. Uh, it isn't to say that they aren't social in some aspect. They're usually found close to one another, and they tolerate, again, one another. Uh, Anyway, so the male copperheads are early to reach sexual maturity, around 2 years, and the females will reach sexual maturity around 3 years old, which of course would probably change depending on habitat, genetics, uh, and things like that. So copperheads will generally mate in the spring, around here, April through May, and they also may mate in the fall as well. They will store the sperm in oviducts, which is what a lot of animals do. Think like ants. Uh, they do a similar thing, which is how a queen ant is able to breed only once and still live 15 years later. She'll be still producing eggs. Uh, of course, when they do this, uh, they'll allow the sperm in at I guess, a time they determine to be right, to allow gestation to begin and to later give live birth, as that is what vipers do in the Viperidae family. Uh, Gestation period is around 100 to 110 days, which is decently long, uh, as far as a reptile goes, especially a smaller animal. And then the gravid female is, as I mentioned, viviparous. She will give birth to around 5 or 6 offspring in a quote-unquote litter. Uh, Speaking of mating habits, the males will fight each other to gain access to females. What this often consists of, especially in cottonmouths, is they'll sort of find each other, and if they know there's a female in the area, they'll sort of slither up to each other. They'll sort of periscope, stand up, and they'll just wrap around each other, roll around, and push each other around. Uh, I don't know how a winner is determined in that case, but there is a winner who is determined. Uh, the other one is going to tuck his tail between his non-existent legs and run away, while the other one presumably gets access to whatever female or females that we're watching. So here we have a broadband copperhead from the Copperhead den, and this individual is foraging for water. Obviously, not every animal is going to find major sources of water like creeks, ponds, inlets, lakes. So in this case, it's finding this in either morning dew or something from rain that is collected on and taking that and sucking it up. Here we have a younger copperhead. This one's going after a cicada. And there's sort of an odd thing that reptiles do, having owned like a bearded dragon, for example, and that they really like looking at food before committing to it. They like it moving. They just, I guess, like knowing it's alive, and that tells them that this is food. This one takes its time, and he finally decides to go in for it. It's going to envenomate it. It will very quickly die from that process, and even if it doesn't, he's got a good grip on it, and he's immediately going to take it down. So, as I mentioned, they prey on a lot of animals, and they will also, despite being generalists, they will still fulfill niches in certain regions. For example, researching this, I found a research paper that I mentioned some copperheads in a region of Colorado. They will almost exclusively eat these red squirrels, and so they end up being the main predator and the main population control of these generally overpopulated red squirrels. So as I mentioned, as you saw, they love cicadas, uh, especially the juveniles, even adults. One second. Even the adults, they will climb trees or whatever. And it's not that it's often that they climb, but they will climb or look around for the cicadas because they absolutely love them. I suppose it's just an easy, very, very easy meal, seeing as they admittedly have the survival skills of a piece of paper. As you saw in the video, they're not really good at running or anything, so it's just an easy, nutrient-dense meal for the snake. Uh, Obviously, they'll eat other reptiles, and amphibians, and they will also eat snakes. They'll eat king snakes, who like to eat them. Uh, and obviously, big thing they're known for is eating your mammals, like your rats, your moles, shrews, just anything that really comes by. Uh, as far as hunting goes, I didn't really put that on here. Generally, all pit vipers are ambushed. They'll sort of sit in an area, wait, and if something comes by, they'll strike it. And instead of like colubrids that will, then either immediately try to swallow it or constrict it to death, the pit viper relies off its venom for hunting. So it'll strike it, and given that these are generally small animals, they'll expire very quickly, and then the snake will follow a scent trail or visual trail to that animal to then ingest it. Obviously, when it comes to animals, you don't just look at what populations they control. You have to look at what also relies on them as a source of sustenance. So obviously, the first one is birds. Your birds of prey, especially. Uh, Snake is a very good nutrient-dense meal, especially vipers, which we'll get into with the next one. For these large mammals, it's just such a great meal for them, and something animals like opossums, and of course in other regions of the world, you have animals that have adapted to withstand the venom of these snakes, so they may be able to better prey on them when they need to. Uh, Obviously, when you have snakes like king snakes that are named so just because they eat other snakes, take king cobras, which are also snakes that eat other snakes, indigo snakes, which specialize in eating again snakes. Uh, There's a obviously advantage to eating snakes when you're a snake and you have to consume a large prey item. You have to consume, sorry, you have to think about which way you're going to eat it, how the limbs are going to fold, this, that, and other. And when you're eating a snake, you don't have to think about that. You just straight down the hatch. And it also just, again, works out as far as vipers go. So many snakes wanting to dedicate to eat vipers. As I mentioned before, something like colubrids, they are generally more active hunters. They tend to be more lean, whereas vipers, being ambush predators, they get all sorts of fat, so they're just full of nutrition for something like a king snake or any other snake that prefers eating snakes. Uh, this is more of a joke. Obviously, humans are large killers of snakes, not necessarily predators, but more of just kill them. For various reasons, some of which are very understandable, given that the copperhead is a copperhead at the end of the day. So I'm going to head and leave y'all with some of my notes over this. There's a research paper, which I'll point at the end. It's very in-depth, makes a lot more sense once you've had things like chemistry, organic chemistry, things like that, physiology, just a lot of classes, and admittedly this is not something I understand terribly well. More or less, this is the effect on the venom, so we will skip through the next few slides because this just goes through it. So your two components of venom in the copperhead, well, the three, but the main two are going to be cytotoxins, those that affect cells and tissues, and hematoxins, those that affect clotting and red blood cells. So the first section is going to be your SVMPs, which are your snake venom metalloproteinases. Wonderful long name. What they're going to do is they're going to break down the structural proteins that make up blood vessels, which will cause capillary walls to rupture and cause some internal bleeding and swelling. As mentioned here, it allows plasma to leak, which is going to cause edema. These also will target collagens and other proteins, which will lead to local tissue necrosis. I forgot to mention these SVMPs will also attack skeletal muscles, which will destroy them as well. Generally, as far as medical advice goes, I say take it from a doctor. You don't have to take it from me. But given all the medical advice that exists from, say, the Red Cross and the CDC about snake bites, the last thing you want to do with a snake bite is to tourniquet it. And this is one of the reasons, especially if the snake has myotoxins, those that will break down muscles, such as the cytotoxins will. what ends up happening as you cut off blood supply is the venom will still do its thing. It has no problem spreading, as venom will generally spread lymphatically, which means a tourniquet does nothing, as it only blocks off arterial and venous flow. So what ends up happening is as these uh, muscles die, these skeletal muscles die, it can lead to rhabdomyolysis. And if this tourniquet is to be taken off after being on for an extended amount of time, you already risk ischemic reperfusion, which is where those deoxygenated or oxygen deprived cells and tissues are already dying and releasing uh, terrible toxins because they're not going under apoptosis. Instead, they're just lysing. Uh, on top of that, again, as mentioned, we have rhabdomyolysis from the muscles, which just given those two alone makes it very dangerous to take off that tourniquet. So, generally, as far as the tourniquet goes, if you apply it on a pit viper bite, not only will you probably lose the limb, but it may kill you as well as soon as that tourniquet is released, which is why doctors should be the only one to release tourniquets. Anyways, I digress. So, talking about the hematoxins, the serine proteinases, the snake venom serine proteinases, which again, if you're ever writing something, double check it because here we have serine proteinases, which is here the SVSP, and these are the metalloproteinases, and I accidentally put that these are SVMPs when they are SVSPs, which are quite different as far as toxins go. One second, let me get rid of this pen. Anyway, so the three main ones we're going to look at as far as hematoxins go, which is a good majority of the Copperhead's venom, at least the eastern Copperhead, is the SVSPs. Sorry, let me go back here. The SVSPs, the phospholipase, and the disintegrins. As far as those who've had basic biology and chemistry, we know that something ending in ACE is an enzyme, and what this is going to do, this phospholipase, is obviously the phospholipid bilayer that surrounds cells. This is going to hydrolyze these bonds, turning them into fatty acids, basically just further breaking down the cells and these disintegrants. So what's unique about SVMPs is they will act like thrombin, which is part of your clotting factor in your platelets. Already before that, these SVMPs, SVSPs, they're already going to basically prevent clotting, but say that a clot is able to form to try and help to stop the bleeding in an organism, especially internally bleeding. These SVMPs, through acting like thrombin, will basically cause any clots that form to be extremely weak and able to break apart. And as I mentioned again, the disintegrins, which is also a very important one to remember for later, they will bind with the integrins on platelets, which again prevents linking through fibrinogen, again preventing any clots forming, and the SPs will prevent those clots that do form from being very strong. So basically, in a lot of animals, it's going to lead to rapid internal bleeding. As far as humans go, this is not a terribly large concern. Of course, here we at the bottom, these phospholipase A2, it again hydrolyzes the bonds and phospholipids, causing the cells to lyse. As I mentioned, as you know from basic biology, cells are supposed to undergo apoptosis, the programmed cell death. And one of the things that happens is they will section off things like lysosomes to prevent toxic things from leaking into the organism or just their surroundings. When a cell lyses, it does not have the chance to do that, so this further increases toxins in the blood. And again, this is all it's mentioned here. If you want to pause it, this is basically just a simplified version. And again, we'll look at the Boykin paper in a little while. Alright, so this is about LD50. If you're ever wondering why PETA hates biologists, this tends to be the reason. LD50 is going to be our lethal dose for 50% of a population, and we measure it in milligrams to a kilogram. And what we do here is we take a test population of an animal, generally mice, sometimes monkeys or other primates, and we give them a bunch of substances. So we give our entire population or at least one of our groups, the same amount, and basically see how much it takes to kill 50% of our population, which tells us the about how toxic a substance is, or just toxin in general. And one study that I reviewed for this found that the eastern copperhead had an LD50 of 10.5 milligrams per kilogram of body weight through intraperitoneal injection. Now, of course, the rest of these are also going to be IP injection to keep the results sort of similar. So here we're going to start off with putting the copperhead on the bottom. And next we do bring up to one of my favorites, the pygmy rattlesnake, which is a native species here in Bowie County. Again, they're not the same as Crotalus rattlesnakes, their own genus. They have these tiny little rattles, and they're small snakes, usually under a foot. Bright, generally have bright orange lines going down them, so they're a beautiful snake. Anyways, they have an LD50 through intraperitoneal injection of close to 7 milligrams a kilogram, so a little more toxic, at least to the test subject of mice, than the copperhead was. We have the relative of the copperhead, the cottonmouth, which had 5.11 milligrams per kilogram of body weight. And finally, at the top of the pit vipers, we have the timber rattlesnake, the canebrake, at 2.04 milligrams per kilogram of body weight. The reason I have had to mention that this is the canebrake is in the past, again, as a lot of other species snakes had, it was considered a separate species from the northern timber rattlesnake. The southern populations were called the canebrakes. I believe the canebrake was considered to be a subspecies of the northern species, and then recent genetic testing, similar to others, we went, oh, this is the same snake, just different area, different look. But the reason I still want to mention this is canebrake is despite the fact that we've discovered that northern and southern populations of the timber are the same snake, it does not change that they have different venom, and that the canebrake tends to have stronger venom, in this case 2.04 milligrams per kilogram. Granted, all of this is relative to us, seeing as we are not mice, and these snakes are generally not going to bite you in your liver area, so this is all just relative. Anyways, this is not the most venomous snake, not even in Bowie County, but in the States. That goes to the Texas coral snake, in a lapid, Micrurus tenor at 0.78 milligrams per kilogram. Granted, as far as the coral snake goes, and danger to humans, coral snakes are fossorial, meaning that they burrow a lot. They are not snakes that humans encounter very often because of that. They tend to generally only come up after rain or wet times. Small snakes, too, they don't have a lot of venom. So if there is one to crown the most dangerous, it will probably be, at least in this area, Bowie County, the timber rattlesnake. They are much larger, they're much more common, they have more venom. So just some quick copperhead myths, something that you may hear passed around a lot. First of all, the really big one is that the baby copperheads, or just juveniles, cannot control their venom, and when they bite something, they will release all of their venom because they cannot control it, which is, of course, not true at all. Just any snake that possesses venom, from the moment they are born, they can control that venom. Sort of think when you came out the wound, if you came out the womb normally, you started to breathe on your own. That is not something you had to learn. That is something that you are instinctually doing. This is true for snakes too. We've tested them, right out the, well, in this case, as far as vipers go, right out the womb, and they will also have control over the venom, choosing how much to inject. If anything, a baby, an infantile or juvenile copperhead is probably less dangerous than an adult because they have less venom since they are smaller. Another one you'll hear is copperheads travel in pairs. Kind of true, but not really at all. Outside of mating, again, I mentioned they're not terribly social. They tend to be somewhat solitary. And just because you see one doesn't mean there's a lot. Although I will say, if you see one, there may be more nearby, not because they associate with each other, more of just because if the conditions are right in the habitat to house one, there's probably more nearby, since again the conditions are right. This one's controversial between a lot of people. I tend to stand with herpetologists who say snakes do not chase people. Generally, you hear this more about cottonmouths, and generally arguing is, well, I've seen my friend Billy Bobby Joe. He got in the water and he walked into a nest of water moccasins, got bit 50 times and died. Uh, I've heard that story a suspicious amount of times. But in this case, again, if you come up on a snake, it is not going to chase you per se. So we're going to leave that as a myth. They are defensive. They have no reason to want to chase you. Think to a grizzly bear, very, very large mammal, very dangerous mammal that is significantly taller and larger than us. I mean, what, are they 12, 15 feet sometimes in larger individuals? That'd be like one of those standing up and you going to go chase it down. That'd be a terribly stupid idea and would probably lead you being killed and therefore, again, not passing down your genes. So these snakes probably have no interest in chasing a person who is 200-300 times larger than they are. Uh, Now, I do believe that they tend to be misinterpreted as chasing. They do have memory, of course, so if they believe that they know an area they can get away and that area happens to be behind you, they will, I guess, go ahead and decide to run in your direction just because they know behind you or around you is something safe they can get around. I've also always had this thought that under certain conditions that a person is to cast a shadow. They may see the shadow and believe that is something that they can get away from you in, and so they'll chase your shadow, hoping that that is one way they can escape you. Another one I've heard is that copperheads can't climb or swim. Obviously, as we discussed earlier, they can climb, and they can also swim very well because all snakes can swim very well. And most of them can also climb very well too. That you have stronger climbers. Take your rat snakes or any snakes in the Pantherophis genus. They're excellent climbers, and that's where they usually get their name. And where a lot of people see them is climbing something, which, as far as Pantherophis obsoletus, the uh, common black rat snake, that tends to lead to it climbing in really weird places. I've heard saying among other herpetologists that if you find a snake, at least in the U.S., in a really weird place, like above a door or on a door or like halfway up a house, and you have to question how did you get there, it's probably a rat snake because they just get in the weird tight spots again to find their food. Uh, Another one I've sort of added to this swim thing is I've heard if a snake swims on top of the water or like sticks its head on top of the water, it's venomous. And if it's under the water and moving super fast, it's non-venomous. This is also false. Copperhead will be on top of the water. Cottonmouth will be on top of the water. It could be under the water. Same with, I guess, copperheads too. Uh, Water snake, vice versa. They can be on top, bottom. It does not really matter. So, as far as being these snakes being useful, I've heard a relatively fair argument against snakes that we don't need anti-venom for snakes if there are no snakes. But of course, these snakes obviously serve other, uses to us outside of anti-venom. So, again, more medical applications. And just something to point out, according to the CDC, about 5 people die every year in the U.S. to snake bites. You genuinely stand a better chance of winning the lottery and or just being struck by lightning than you do dying from a snake. Treatment's readily available. Outside of obviously allergic reactions, immediate fatalities rate, you generally have hours, or like even in case of a coral snake, which can take a day or two for the neurotoxic venom to set in, you could have a day or two, to get to a hospital, to get treated, and whatnot. Take an animal like a dog, I believe 70 to 100 fatalities occur per year, according to the CDC. That is a significant more dangerous animal. And of course, uh, us being in biology, we have to know statistics. We have to consider something like, an argument for it against that is, of course, dogs kill more people because people are around dogs more often than they are snakes. But I'll also argue if you've been outside, you've been around a snake, whether or not you knew it, they were probably there. Even someone like me who goes to actively look for them, we miss a lot. They're well camouflaged. They know when to sit still. So chances are you yourself have been very close to a snake, whether or not you knew it very recently too. Uh, Speaking of death, cancer, at least this year, is estimated to kill close to 620,000 people. If you ask me, one is a lot more dangerous than the other. So in the early 2000s, researchers looked at all sorts of snakes, including copperheads, and especially with copperheads, what their venom could do to combat common diseases. In this case, cancer. What was discovered is in the proteins of an eastern copperhead, and you can see one down here that I photographed. Also, something I want to point out. It's not going to be terribly visible, and I wish I'd talked about this earlier. As I mentioned, triangular head, it's generally a feature, but you can find some snakes that don't. But another one you hear is that one way to identify a viper is that they have cat eyes, sort of the slit pupils. And that is true. That is something that is generally a defining feature of them, given their lifestyle. But first of all, other snakes, given like boas and pythons, they have slit eyes, and there's even colubrids, like the cat-eyed snake, which has slit pupils. In this case, you can look at this female individual who was recently gravid and given birth, if you want to wonder why she's so large. You look at her eyes and you'll notice they are wide. I took a picture of her with a flash on in the dark. Under dark conditions or even under just any conditions, they will have round eyes, just as most other animals have. So, going off pupils is not a generally reliable thing on determining whether or not something is venomous, as far as a snake goes. Anyway, so in their venom, it was found that they have a protein, sort of disintegrin, as we mentioned earlier, called contortastatin. Uh, and when it was tested, they found that it had some effects on tumors. So what was done later is it was taken the genetic sequence in the copperhead that codes for the production of that disintegrin was taken, and it was combined with ecostatin from saw-scaled vipers, which right here. I believe these are Middle Eastern, but I'm not terribly familiar with them, to be honest. Uh, the the part that also codes for their production of ecostatin. It was combined, and these genes were put into E. coli, which then produced the protein by themselves, vicrostatin. This is also sort of thing. If vicrostatin is to uh, become a main treatment, since it is still in trial, obviously, we can't just have snake farms. I mean, we could have snake farms, but it'd be terribly cruel and hard to manage, and of course, this could increase the incidence of bites. Uh, so this is one way to get around things like this: is you can have E. coli or some other bacteria produce these proteins for you through something like CRISPR, gene editing. As I mentioned, vicrostatin or just contortastatin, both, they've been shown to inhibit the spread of breast, ovarian, and prostate cancers when tested with mice in cell culture. Of course, when it comes to any medication, there's a lot of trials that they have to go through to make sure that this is safe for people. Because it really sucked to give this to somebody and have some terrible side effect that could have been avoided with thorough testing. As far as like coexistent go, it doesn't mean you go outside and frolic with them. I do not recommend that at all. Uh, as interesting as these animals are, they are still pose a medical danger to humans, so they should be admired at a distance. Anyway, so both species of the copperhead, at least in Texas, are least vulnerable. They are not, at least in the current time, close to any sort of danger of extinction or local extinction. I believe there's a few states in the sort of Appalachia area where copperheads are considered endangered and efforts are being made towards them. I believe in maybe Maryland. Not quite sure at this moment. Anyways, my thought is that, as we continue urban sprawl and these conflict zones get hotter where people kill snakes, this could become also habitat fragmentation. These snakes require, you'd be surprised how much they require to actually live. And if you have fragmented habitat, like for example, behind my house, we have about uh, half an acre of woods, and there are no copperheads there because it is not something that can support their population. Uh, but also through something like road mortalities, where they're hit at, especially at night when they're traveling during the summer. There's just a lot of issues that I believe could, in the future, maybe even the soon future, a decade or two, lead to a concern for their population. Just some general advice as far as these snakes go and what people can do or what even you can do. First of all, I've heard a lot, like I recently was told by someone I work with, that she puts mothballs down to deter the snakes, and that will not work. That could also possibly, in fact, attract them as by putting mothballs down. While that will detract some insects will find that and use it as a place to hide. And then there could be other animals that eat those insects that come for those insects, and then whatever eats insects, snake won't be far behind. So the whole food chain could be just sitting there waiting for it. But again, there's no natural chemical deterrents. You can't spray this oil or plant this plant to keep them away. They don't care, to be frank. So it's really just a waste of time. The only deterrent, I've heard of a rattlesnake gate being developed, but outside of that, there's not really much you can do as far as that goes. But what you can do is eliminate some of the things they need to actually live. Uh, so keep your yard or just anything in general clutter free. You know, outside of things they could hide, think of things laying on the ground, tall grass, wood, rocks, uh, pots, anything like that that they could get under and use as shelter. Uh, if you have body of water, which they will sometimes relate to, not necessarily as much as something like the cottonmouth or a water snake, but just as a source of water and or food. Obviously that attracts amphibians and other reptiles and other animals. Keep the surrounding vegetation around the body of water low, and any emergent vegetation by the bank low is again just because you've removed that vegetation that may keep other animals out and the copperheads from hiding. If you have emergent vegetation, that's still going to attract amphibians and other animals, which again, they will follow the food chain. Obviously, if you would like to really pull out your inner Disney princess and you want to make your habitat more uh, habitable for snakes, then do the opposite, leave stuff around. I will say something that snakes really like is tin sheet. I don't know why they like it. Maybe it just the fact that it retains heat. Maybe that it's easy to get under, but they are just huge fans of it. So generally with snakes, again, especially venomous snakes, you don't want to handle them or really get near them. Uh, but if one were to run across, you can generally leave them alone and they will leave. Especially if they've seen you. Once they see you and you leave, uh, again, something I forgot to mention is take other vipers. Your rattlesnakes are known for rattling. That's one of the defense responses, is they will stand up to you. Generally, of course, it's not guaranteed. A lot of rattlesnakes actually do not rattle and will try to remain hidden and rely on their camouflage. The cottonmouth, again, what it's named after is throwing that, opening its mouth agape and throwing that defensive display. Uh, so they will also sort of stand up, but copperheads, their main defense is to sit still and hope that you do not see them. They will freeze, and that's also what leads to them being one of the more common bites, is instead of making themselves known, they'll sit there and you may accidentally step on them, in which case they will decide to try and defend themselves. Uh, but anyways, again, if they see you, they're going to freeze up, and if you leave, they're probably going to take that opportunity to run. I've seen that happen with a lot of snakes. I know they'll freeze up. I'll turn around to go grab like a snake hook or something, and 10 seconds later, they're nowhere to be found. Uh, Alternatively, if you really need them gone, you could like take a water hose to spray them. If you put it on jet mode, trust me, they're not going to be happy with being soaked, just as any animal or even person would be. You could also long object like a broom carefully, just sort of shove them to the side, and then be like, "Fine, I'll go." Of course, you have a snake hook. Be careful with that. Usually, if you use a snake hook, you know how to use one, so not too much advice to give there. Obviously, you should, if you're going to mess with snakes, you really need to know what you're doing because it can be dangerous, and even experienced people still get bit. And just be careful. Also, we have snake tongs. Although I will say, as far as they go, you see them with their little metal teeth. That's not really good for the snake to clamp down real tight. It can break ribs, fracture bones, damage internal organs. So the best thing to do, and a lot of professionals who use these tongs, especially for extended period of time, is to cover the teeth up, to add some sort of padding to prevent damage to the snake. And as mentioned here, we finally get to one of our sisterous snakes. Cistrus cataneus, which is the eastern Massasauga found in about the Midwest to sort of East Coast-ish area. It has a weird range, but it's a beautiful, tiny rattlesnake. Uh, if you're going into an area that you suspect many snakes may be present, I would say get something like snake boots. I personally prefer gaiters, just because of their coverage. Granted, no boot, no chap, no gator is 100% snake proof. It's more of just an extra preventative. The easiest way to prevent, being bit by a snake in an area that you think that they are is watch where you're stepping. Don't step over something. Don't reach into something. The less you can put yourself around where you think a snake may be, the better. Just pay attention with what you're doing. In general, that is the best defense. Something I'll also say is like boots. Not necessarily steel toe boots. Obviously, they're not going through a steel toe, but any part of the boot, even like a nice leather boot. Even something like Copperhead, they have a very impressive bite force. They can get through that leather of the boot. So that is not something that's going to help you. But again, nothing is 100% snake proof, more or less, unless you want to wear knight's armor out in the field. Uh, but just pay attention where you are, and that's one of the best ways to prevent a bite. Uh, as far as Texas goes, it's unfortunately not illegal to just go out and kill snakes, outside of the timber rattlesnake, and of course, once you head into South Texas, you start seeing snakes like the indigo snake, which are federally protected, which are very, very illegal to kill. But snakes like states like Arkansas, they have very strong laws against killing snakes unless it poses an immediate danger to yours or someone else's or a pet's life. And this is, there are some big fines, especially with protected snakes. You could even face jail time for killing a protected snake. So obviously, if you don't have to kill it, there's no reason in killing it. They have, just as much right to live, and this is a lot of times where you find them is in conflict zones, urban conflict zones, especially in newly developed areas close to where their habitat was. That was their habitat to begin with, so just have some empathy for them if you would. Just a quick little thank you if you'd like to look at more broadbands. This woman's wonderful. She posts all about these broadbands that she has been sort of allowing to live on a property and prosper, and gives them all sorts of wonderful areas to live. This is a sort of friend here in Texarkana. She has a collection of vipers here. The first one is Tremacerius insularis, the blue insularis. Beautiful snake from Asia. And they get decently big. This individual is about a foot long, and they're just this wonderful blue. Here's that same African bush viper from the slide about the Selenogoliphus fangs. This time he is a little less aggressive with us. Pretty little snake. This is a western diamondback rattlesnake. Of course, you do not notice the general tan patterns in the diamondbacks because this is an individual whose parents were an albino and a leucistic, and this specific color pattern is called a buttermilk. Something I'd like to point out is if you notice, all these pictures are pretty clear of these snakes, right? These snakes are used to humans. I was able to get my camera a few feet away from them and take these pictures. If you notice very faintly here, you will see my reflection, because this is not a snake you want to take the glass off of, because they tend to be very striky, and they can strike a fair distance as well. This individual, as you see, this female, she was coiled up and sort of just lightly rattling at me, not in a full-blown, coil up like they usually do and sort of stand up, but she was just more of like I don't really feel like being bothered right now. And this one right here is a speckled rattlesnake, I believe found in the sort of western regions of the country, I think like Arizona and those sort of areas. Very tiny little rattlesnake, and this one did not mind my company as much, and so I was able to get some better photos of it. So these are some of my sources in case you want to do some more looking into these snakes. I did say I'd try to point out specific. One second, see if I can't find it. Well, it may not be here right now, I guess I'd forgot to put that up in my sources, but if again we go all the way back to these toxins, uh, roll all the way back real quick. Where is that? Citation. Again, Boykin et al. Usually, if you look up Copperhead, Boykin et al., it should pop up. It's called uh, Proteomic Analysis of a Cystrogen Contortix Venom using 2D Electrophoresis. It's a very interesting read. I remember, showing this paper off while researching this presentation, and these people would look at it, these big biology nerds, and they'd of course think it's cool, and then they really get to reading it, and the venom is surprisingly complex. There's a lot of components. These components have varying effects, and these of course depend on population, region. It's a very interesting read in case you want to learn more, especially go in depth into more of the biochemistry of these snakes. And of course, these are some image references. About half of them I photographed, or the Copperhead Den photographed, and I was allowed to use. A lot of commons, and we have two that were AI generated. The picture of the Howard Glory moccasin, which is terribly difficult to find, so unfortunately that's what I had to resort to try to get some sort of visual representation. And that picture of the Hershey Kiss, just because I couldn't find a great one that, I had rights to use. But anyways, that is, the presentation. I'm glad you listened, and hopefully you learned something from this.